Hello, everyone. It's kind of strange because I can't see you all, but hello, good morning, good afternoon, and good evening to you all. We have so many people joining us today from all over the world. Thank you for setting aside time to learn more about Roche and Genentech's Angelman's program. We understand that these are unprecedented times we are all facing with the COVID pandemic, and then most recently, heightened awareness of the Black Lives Movement, or the Black Lives Matter movement, which is an international human rights movement. So we know that it's a heavy time, and we know that there are so many other things that, that may need your attention. So we thank you again for joining us today, and we hope that you find today's presentation helpful to you and your families. So our goal today is to inform you about Roche's Angelman Syndrome program, and we want to thank Angelman Syndrome Foundation for making this request to learn more about our program and also hosting us today. All right, so without further ado, I'm going to get started with the slides. So my name is Zena Chi. I'm with Patient Advocacy Relations with Genentech. Genentech is the U.S. advocate, excuse me, got my daughter in the background. Genentech is the U.S. affiliate of Roche. And so we are one company, and I would like to hand it over to my colleagues, Shady and Brenda, to do their introductions.
Thank you, Zena. I'm Shady Sidholm, the Global Patient Partnership Director, working in Roche, based in Switzerland, and I'm working with focus on Angelman.
Hi, good morning, good afternoon, good evening, everyone. My name is Brenda Vincenzi, and I'm the new medical lead for the Angelman Syndrome program, taking over from Michelle Krishnan.
Thank you, Shady and Brenda. Oops. Okay, so today's agenda, we've already done our introductions as Roche and Genentech, and we will go into our Angelman Syndrome program history, and then we will also talk about Freesias, our observational study that is currently ongoing and recruiting, and the big announcement, which is our phase 1 Tangelo study. We will introduce this, the details, and talk about the Roche molecule. And then we will open it up for Q&A. So as far as Roche and Genentech, as I mentioned, we are one company, and our mission is to transform the lives of people with serious diseases. And just to let you all know as well, this webinar will be recorded. So if you miss any portion of it, or you have friends and family that want to listen in at another time, it will be housed on the clinical trials website through Angelman Syndrome, the Angelman Syndrome clinical trials website. So it is recorded, sorry. So anyway, Genentech and Roche's mission is to transform the lives of people with serious diseases. Together, Roche and Genentech has had 38 FDA-approved medicines. 22 of them have been breakthrough designations. And so we've been in the Angelman syndrome space for 6 years in partnership with many organizations and researchers. And so Roche and Genentech has had a long-standing commitment in rare diseases, starting back from, growth hormone supplement, Neutropin. This is the FDA's, actually the first approved, Genentech's first approved growth hormone product, and this was back in 1985. And to most recently, I think Hemlibra and Esbriet probably are the ones that stand out. But you can see that we have a commitment to the rare disease space. And we do this through, I will pass it on to my colleague, Shady.
Thank you, Zena. I would like to say, yes, we have a long history with rare disease, and we do this to develop breakthrough treatments for people living with fatal or life-limiting rare disease. And we do this based on 3 main pillars: follow the science, achieve deep understanding of the disease, and form a strong collaboration with the community. Next slide, please. With regards to Angelman, we started in 2015 listening carefully to the community, attending many congresses such as the ASF Congress and FAST Conference. And then in 2017, together with ABOM, we created the disease concept model, which our scientists use to create our molecule. And then in 2018, Freesias study was co-created with family and scientific community. It's in partnership with Ionis and Biogen. September last year, we started recruiting patients for Freesias, and recruitment of Freesias was a little bit delayed due to the COVID situation. But at the same time, we were working on designing the Tangelo, which is phase 1 trial in collaboration with the patient community. And we hope that in this summer, this is our target, that we will continue recruiting for Freesias, and we will have our first patient in for Tangelo study. Next slide, please.
Shady, I'm not sure. Is everyone seeing us right side up? I think someone mentioned that you were upside down. You look right side up to me, but can everybody else see us okay and hear us okay? If you want to type the answer into the chat box. Okay, people are saying we look all right. Okay, good. Sorry, Shady.
Good. I started to get worried that
upside down the whole time would be pretty funny.
Yeah, but I just wanted to say then that that was only possible through collaboration with the Angelman specialist advocacy group, patient families, sponsors, scientists. And here I would like to thank all of you who were part of this journey and still part of our journey. And here I would like to hand it over to Dr. Brenda to give you more information about both studies, Freesias and Tangelo.
Thank you, Shady. And once again, good morning, good afternoon, good evening, everyone. For people that might have joined late, so my name is Brenda Vincenzi, and I'm the new medical lead for the Angelman Syndrome program, taking over from Michelle Krishnan. So today, of course, as mentioned by my colleagues, we are extremely excited because finally we can share some details about our molecule, UB3A-LNA, and our phase 1 study, Tangelo. But before I do so, I want to introduce you very briefly our Freesias study, our observational study. So I will give you an introduction and a brief update. Next slide, please. A brief disclaimer: any opinions given today are my own. Next slide. So moving from a scientific concept into clinical trials in Angelman syndrome requires a very strict collaboration between patients, caregivers, families, academia, so scientific investigators and clinicians, industry and regulatory authorities. And the ones that you see on the left side of the slide are just a few of the very important questions that together we need to answer before and during the clinical trial. So collaboration is key. Next slide, please. One example of collaboration is the one with ABOM. So we ask the clinicians and caregivers to answer a very important question. What are the most important concepts of interest for clinical trials in Angelman syndrome? And we received a consistent picture. So the ones that you see on the right side are the most important concept, and we listened to this and we incorporated this concept into our our current clinical trials and going forward into our clinical planning. Next slide. So Freesias is our observational study. And there are different observational studies, and they're very important to guide clinical trials. In particular, Freesias can help us to, in order to design the clinical trial, but also to play a role as a control cohort. So like a comparator for next drug studies to potentially reduce the placebo numbers and the length of placebo. Next slide. So as mentioned, Freesias is our observational study, meaning that there is no drug involved. And the study sponsor are Roche and Genentech, and is co-founded with Biogen and Ionis. Next slide. So Freesias has a characteristic decentralized design, meaning that, as you can see from the slide, there are only 2 in-clinic visits. The rest of the assessments are done at home, at the child's home. The primary objectives are the feasibility and value of novel endpoints and biomarkers. Of note, Freesias is only running in the US. Next slide, please. This is an example of feasibility in Freesias, in particular on the digital biomarkers. And on this slide, you see listed the overnight home sleep EEG, the sleep mat, and the e-diaries. So a technician will go to the child's home and will place the EEG so that the child can sleep in his or her own bed while we record the EEG. The sleep mat is a completely passive digital biomarker. So the family doesn't have to do anything apart from placing this mat underneath the mattress. And we can record the important data on heart and respiratory rate and activity. The sleep and the seizure diaries are e-diaries that the caregiver will fill out. And this will give us some important information about sleep and seizures. Next slide, please. So, as you know, COVID-19 has put on hold the majority of clinical trials around the world, including Freesias. But we are excited to say that we are almost there. We are almost ready to start to go back to full activities very soon. And we want to thank all the families who have participated in the study so far. Next slide. And also, we want to expand our thank you to everyone involved, all trial site teams and study coordinators, academic collaborators and study investigators, and many more that cannot be listed on this slide. So these are things to be aware of. Freesias, as mentioned, is a non-drug study in collaboration with Roche, Ionis, and Biogen. These 3 are separate companies working on drug studies for Angelman syndrome. Roche is working independently, so we are working on our drug study called Tangelo. Ionis and Biogen are working together, and they are working on their Ionis Biogen's drug study. Genentech is the US affiliate of Roche. So eligibility for any future drug studies will be based on specific criteria. Freesias participants that met specific conditions will have priority over non-Freesias individuals to be screened at a study center. They will receive a one-time priority screening voucher, which can be used in either of the 2 drug studies, either with Roche or with Ionis Biogen. But please note that participation in Freesias does not guarantee automatic eligibility for any future drug study sponsored by either Roche or Ionis Biogen. Next slide, please. And now that I gave you an introduction and a brief update about Freesias, I'm going to introduce our molecule, UB3A-LNA, and our phase 1 study, Tangelo. Next slide, please. So here you see the steps in drug development. And in the middle of the slide, in orange, you see where we are today. So today we are about to start what we call clinical development. So we are about to start phase 1 and our Tangelo study, which means that we are going to start a phase 1 into humans for the first time. But what we want you to understand is that so far there's been a lot of work at Roche for our discovery group, our preclinical groups, and we initially identified a target, the protein target, and then more than 2,300 molecules were tested, and the most potent and safer was chosen. Next slide, please. And here is our drug. So it's an antisense oligonucleotide, or ASO, that for simplicity we call LNA, simply because it's being chemically modified with a locked nucleic acid, or LNA. So our LNA acts on the UB3A gene by producing the missing UB3A protein in Angelman syndrome. Next slide, please. And here is the mechanism of action. So as we know, everyone has 2 copies of the UB3A gene. 1 copy is from the mother and 1 copy from the father. Both gene copies are active in most parts of the body, but in neurons, only the mother's copy is active. And please refer to the picture in the middle. Thank you. So the father's gene is fine, is there and is fine, but is silenced by an RNA called the UB3A antisense. So that in typically developing individual, the protein, the UB3A protein, is only produced by the maternal allele. In Angelman syndrome, what I just described for the paternal allele is mirroring the situation. So the paternal allele is silenced, is not producing any protein. The maternal one, for different reasons, is not producing the protein. So the result is that in Angelman syndrome, we don't have production of functional protein in neurons. And this on the right side, you see what the target of our treatment. So please refer to the circle in red. You see our LNA, so our drug is going to bind to this long antisense sequence that is responsible for stopping the expression of the protein from the paternal allele. So what the LNA does is to remove the brake so that the paternal allele can start expressing and producing the protein that is missing in neurons. And I want to point out that we look at our LNA as a disease-modifying therapy. Next slide, please. And here is Tangelo. So Tangelo is our phase 1 study. It's an open label, meaning that there is no placebo, no randomization. Everyone enrolled will receive the drug. It's a multi-center study, so we have different centers in the US and outside the US to investigate safety, tolerability of our compound, and what we call pharmacokinetics and pharmacodynamics, meaning that we want to understand what the drug does to the body and what the body does to the drug. Next slide, please. And here is probably the details that everyone is looking for, and we are very excited to be able to share some of the characteristics of Tangelo. So we will enroll up to 66 individuals with Angelman syndrome, both males and females. The age goes from 1 to 12. The genotypes included are mutations and deletions. And the location is going to be 5 to 6 countries, so the US and countries outside the US, for a total of 17 sites. The study duration is up to 58 weeks, so we aim to start in the summer of 2020, so very soon, and to end the study at the end of 2022. Next slide, please. Here is the study design. So in orange on the left side, you see the screening time. So we have a screening window that goes up to 6 weeks. After that, we enter the treatment period in light red. So we start with the first dose. After 4 weeks, we have the second dose, and after other 4 weeks, the third dose. So the medication is administered intrathecally. So there is a lumbar puncture to make sure that the medication reaches the brain. After these 8 weeks of treatment period, we'll enter a follow-up safety period up to 44 weeks. The total duration of the trial is up to 58 weeks. So just a summary, it's an open label, so there is no placebo. Everyone will receive the drug. Multi-center study. You saw there are 17 centers. With a flexible, adaptive, multiple ascending intra-participant dose escalation. Once again, it's an intrathecal administration. The objectives are safety, tolerability, pharmacokinetics, and exploratory pharmacodynamics and efficacy. Next slide, please. So before we go to the Q&A session, I want to spend just a couple of minutes, just 2 slides, on very important terms in clinical trials. So before a participant can enroll in a clinical trial, they have to give their informed consent. In Tangelo, the informed consent will be signed by the caregiver or the legal representative. In Tangelo, we will also have in place a very informative and easy to understand video on the informed consent. Only after the informed consent has been signed, participants will be able to be recruited, screened, and if they are eligible, they will enroll or take part into the study. Inclusion criteria are the factors that allow someone to take part in a clinical trial. Exclusion criteria are the factors that prevent someone from participating in the trial. Next slide, please. So people who are interested in participating in a trial, we go through a screening process to check if they meet all of the inclusion and none of the exclusion criteria for that particular trial. If after screening they meet all of the inclusion and none of the exclusion criteria for that trial, they will be eligible to join the study. Enrolling is the process of entering a person into a clinical trial. Once a person has been enrolled, the participant will then follow the clinical trial protocol. A person can only be enrolled if there is an open slot for them in that study. Of note, a person can leave the study at any time during the trial. Next slide, please. So now I hand over to my colleagues, to Zina and Shady, for the Q&A session. So thank you very much for your attention.
Thank you, Brenda. So at this time, we are going to open it for Q&A. If you do have questions, you can type them into the chat box. We want to mention that we may not answer all of the questions that come through the chat box, but we are collecting it and we will figure out a way to answer if we can. If we can't answer it, it's because the information is not ready to be shared yet, since we're still finalizing some details. Okay, so the first question is, what are the exclusion criteria in Tangelo?
Thank you, Zina. So participants are not able to join the study, the Tangelo study, if one or more exclusion criteria apply, or if not all of the inclusion criteria are met. These listed here are just a few of the exclusion criteria for Tangelo. For example, having a molecular diagnosis different than deletion or mutation, having abnormal laboratory findings at screening, or anything in the medical history or ongoing conditions, so the past medical history of the current condition, that might interfere with the evaluation of Angelman syndrome or with the intrathecal administration of the drug. Or for example, being enrolled in another drug trial.
Thank you, Brenda. And we can go to the next question. If my child is currently enrolled in another clinical trial, how long does he or she have to wait before being considered for enrollment in Tangelo?
Thank you, Shady. Yeah, so this question is related to the previous question. And so participation in another clinical study must be approved by the Roche medical monitor. After the discretion of the sponsor and after discussing with the principal investigator at your site, participant might enroll into non-drug observational studies. In case the participant received another investigational drug, he or she will have to wait some time before they can be part of Tangelo, as is necessary to wait for the body to get rid of the drug. And this waiting time depends on the characteristics of the investigational drug itself and will be evaluated by the sponsor medical monitor, so by us, by Roche.
Thank you. Okay, the next question is, what about other genotypes? Will they be part of future clinical trials?
Thank you. And I understand that I'm sure that online we have families that might feel like left out because of course I mentioned that the genotypes included in Tangelo are deletion and mutation, and this is one of the inclusion criteria. But there is a reason for that, and as you can see on the slides, there is a potential risk of overexpression of the UB3A protein, which means like too much protein in case of other genotypes. So we are working on that, and at the moment we want to gather more data before considering the inclusion of different genotypes, different from deletion and mutation, in future Roche clinical trials. So as mentioned, the inclusion and exclusion criteria of future Roche clinical trials in Angelman syndrome might differ from the ones in Tangelo, and might actually in the future include different genotypes.
Thank you, Brenda. We go to the next question. It's about the age group, and we know that you mentioned that the age group for Tangelo is between 1 to 12 years old. Will adults with Angelman syndrome benefit from the trial?
Thank you, Shady. And this is, I want to repeat what I just said about the genotypes. I understand that we have, I'm sure we have online families of individuals with Angelman that don't meet this criteria, this age criteria between 1 and 12 years of age. And so we have animal studies that have shown that the impact of a treatment could have different effects when provided to younger or older animals. So we currently don't know how this works and is translatable in humans. This makes the study, the Tangelo study, extremely important, as the learnings will be used to provide awful treatment opportunities to patients of all ages and with all genotypes. So our target is to bring a molecule that can be helpful for patients with all ages, all ages and all genotypes.
Thank you, Brenda. And the next question is, can I participate in phases 2 and 3 if I do not participate in phase 1, Tangelo?
Thank you, Zina. So the short question is yes. So participants that will meet all of the inclusion and none of the exclusion criteria of any Roche future clinical trials in Angelman syndrome will be eligible to participate. So inclusion and exclusion criteria of Roche future trials in Angelman syndrome might be different than the Tangelo's criteria and have currently not been determined yet.
Thank you. Next question. Will I be reimbursed for the participating in Tangelo?
Yeah, so let me start by saying that you will not be charged for the study drug while your child is participating in the study. Also, all procedures that are required only for the study and they are not part of your child's regular medical care will be provided to you at no charge. You will also be reimbursed for reasonable costs, for example, transportation, parking, overnight stay at a hotel nearby the clinic, to travel from your home to the study site for your study visit, or to allow for completion of clinical visits.
Thank you, and thank you, everyone. I can see tons of questions coming in, so thank you for your participation. The next question is, what is the impact of COVID-19 on the trial?
Yeah, so as I already mentioned, unfortunately, COVID has impacted a large number of clinical trials around the world, including, for example, Freesias. So, and now it's hard to predict if and how COVID will impact Tangelo, as the situation might change quickly and might also be different from country to country and from site to site. So we are currently planning to start the Tangelo study in the summer without any delay. But Roche will always put the safety of individuals with Angelman syndrome and their families first, and we are in close contact with the sites and the investigators to continually evaluate how the situation evolves and to act appropriately.
Thanks, Brenda.
Thank you. Yes, patient safety first.
Exactly. And Freesias being a US only trial, of course, you know, based on state and even sometimes local government regulations. I know we're still at stay at home, but you know, many states are opening back up. So it will depend on your site, but we are also eager to restart as soon as we can.
Thank you, Dina, for mentioning that. Another very important question, why do researchers or you believe the intervention being tested might be effective? Why might not be effective, and has it been tested before?
Thank you, Shady. Yeah, this is of course a very important question. So as I mentioned during the presentation earlier, the mechanism of action of our drug that we call LNA has the potential of modifying the core symptoms of Angelman syndrome by restoring the production of the UB3A protein that is missing in the neurons of individuals with Angelman syndrome. So indeed, previous studies in vitro, so in cells, and in vivo animal models, show that the reinstatement, so bringing back this protein, this missing protein, translates into rescue of the phenotype. This intervention has not been tested in humans before. And although we don't have any reasons to believe that it might not be effective in humans, we cannot predict with absolute certainty that what was observed in the animal models will be fully translatable in humans.
Thank you. Yes, and I can, let's get to the next question. All right, the next question is, which Angelman symptoms will this drug address?
Thank you. So this is connected to my previous answer and question. So our UB3A LNA, our drug, binds to this long antisense RNA that keeps the father's allele silenced. So it's like a brake. It removes this brake, this block, so that the father's copy can start producing the missing protein in the brain of individuals with Angelman syndrome. So the UB3A LNA has the potential to be disease modifying, which means that it can potentially address all symptoms of Angelman syndrome. This is different from asymptomatic treatment, for example, medication to control seizures or to improve sleep.
Thank you. And I just want to mention that these questions are amazing. Thank you. Keep sending them. And again, anything we can't answer, you're giving us a lot of thought. So we do appreciate it. And if we can't answer your question today, it is being brought back to the clinical team so that we can take into consideration your comments. Okay. Here you go, Shady.
Yeah, so another question, for families that are not involved in Freesias or in the upcoming trial Tangelo, how can we stay up to date with what is happening? And here I would like to thank ASF and many other organizations such as Angelman Syndrome Alliance and the Italian organization for sharing their interest to know more about Tangelo, and for ASF for requesting and hosting this webinar.
Yes, I echo, Shadi, what you just said. And thank you for this question. Of course, we understand that communication with the community is key. And we want to reassure you that we are currently working with our Roche scientific communication group to map our plan for an effective communication so that information will be easily accessible to everyone, regardless if you are a participant in Freesias or in Tangelo or both studies, or if you are not a participant at all.
Thank you. All right. Oops, let's back up a bit. All right, so some of you are asking, where can we get the information? How do I get more information about these clinical trials? And I kind of alluded to the answer already. Shady, would you like to help us answer that?
Yes, yes, please. So very soon the protocol, and if you can share next slide, please, Zina. Tangelo protocol will be hosted on ct.gov, which is, I believe most of you know it. It's a governmental website hosting the protocols for the clinical trials. And after that, the protocol will also be hosted on Roche special website, which is called forpatients.roche.com. And we aim to have the protocol in both websites by end of next week. We cannot promise that. ct.gov, it's not our website, so we don't have control of it, but that's the plan. Thank you.
Great, thank you, Shady. We will keep the patient advocacy groups abreast on like when that posting will be though. So they should be able to share that information with you as well. Okay, so another question. Oh, sorry. Go ahead, Shady. You were going to say something?
No, please. Yeah.
Is it my turn or yours? Okay, let's see. I will pull this one. Okay. Oh, what is the duration of the trial?
Thank you, Zina. Yeah, so I mentioned during the study design slide that the trial's duration is up to 58 weeks in total. So up to 6 weeks of screening, the first phase, and then 8 weeks of treatment, and then up to 44 weeks of safety follow-up with a mandatory safety follow-up of 3 months. So the total study design is up to 58 weeks.
Thank you. Another question, are there any side effects from this investigational therapy?
Thank you, Shed. It is, of course, a very important question. So as mentioned during the drug development slide, if you remember, I mentioned that we have selected the most potent, but also the safer LNA, the safer molecule that we want to bring to the clinic. And in animal experiments, our drug was safe and was well tolerated, so that we don't expect any significant side effect from the drug. Of course, it goes without saying that with any other clinical trial, we will monitor any potential side effects during the trial.
Great. The next question is about individuals with a complicated health history, but is now healthy. Will they be eligible for this trial?
Thank you. So, yeah, this is a difficult question, and in the sense that is not easy to answer without like having more details. So in order to answer, I will need for sure more information. But if your child meet all of the inclusion and none of the exclusion criteria, then he or she will be eligible to participate. And so the answer we will need to evaluate further details, but for sure, if there were some past medical complications and they are resolved, that will be for sure further evaluated by your investigator and by us before we make a final decision. And the reasons are simple, as we want to protect the child. And of course, we will also want to be able to measure any potential effect without confound factors.
Thank you for clarifying
that. Thank you. Yeah. Another question about both studies. Can my child be in both Freesias and Tangelo at the same time?
Okay, so the short answer is no. So your child will have to basically stop the Freesias study in order to enroll in Tangelo. The criteria, of course, are different. For enrollee are different. So being in Freesias, as I mentioned before, does not guarantee an automatic eligibility and enrollment in Tangelo. So in order to take advantage of the priority voucher, your child will have to have at least 6 months of data from Freesias, from being in Freesias, and be actively enrolled in Freesias, or have 12 months of data from Freesias.
Thank you, Brenda. Okay. Let's see. So what are the primary endpoints for efficacy?
Okay, so I mentioned during the presentation that we call efficacy as exploratory. So efficacy in Tangelo being exploratory, and we are currently don't have a predefined primary efficacy endpoint for Tangelo. So Tangelo study together with Freesias, so Tangelo and the observational study will contribute to develop and define endpoints for the upcoming drug studies in Angelman syndrome. So for the future clinical trials in Angelman.
Thank you, Brenda.
Thank you. Thank you, Brenda. And also thank you, Zina, and thanks for everyone. I know you submitted many questions. We try to answer as many as possible. So thank you very much. As already mentioned in the beginning, that this webinar is recorded and it's going to be hosted at the Angelman Syndrome Foundation clinical trial website. So if you missed anything, please check the recording there. And again, I would like to thank all the organization, Angelman Syndrome Foundation, FAST, ASA, and other local organizations for keeping asking about our program and their interest to know about our program. And we would like to always be connected with you. Thank you, and have a good day.
Thank you.